Today, an FDA approval in chronic lymphocytic leukemia, an application accepted for a biosimilar, promising findings in a prostate cancer trial, disappointing data in a melanoma study, and European approvals in head and neck cancer and multiple myeloma. Welcome to OncLive News Network. I'm Kristi Rosa. The FDA has approved acalabrutinib for the treatment of adult patients with CLL or small lymphocytic lymphoma. The approval, which was done under the FDA's Project Orbis initiative, was based on data from two phase 3 randomized trials, Elevate-TN and Ascend. In the Elevate-TN trial, median progression-free survival was not yet reached with acalabrutinib in combination with obinutuzumab or acalabrutinib alone versus 22.6 months with chlorambucil and obinutuzumab. Data from the Ascend trial showed that at a median follow-up of 16.1 months, the median PFS was not reached with single-agent acalabrutinib compared with 16.5 months with rituximab in combination with either idelalisib or bendamustine. This translated to a 69% reduction in the risk of progression or death. Acalabrutinib is also approved by the FDA for the treatment of patients with relapsed refractory mantle cell lymphoma. The FDA has accepted a biologics license application for the proposed bevacizumab biosimilar SB8. Phase 3 findings for SB8 were presented at the 2019 ESMO Congress, in which the biosimilar demonstrated similar efficacy in terms of best overall response rate-risk ratio compared with reference bevacizumab in patients with metastatic or recurrent non-squamous non-small cell lung cancer. Results specifically showed that in the full analysis set, the best ORR was 47.6% and 42.8% with SB8 and reference bevacizumab, respectively. The risk ratio was 1.1, which was within the predefined equivalence margin. In the per-protocol set, the best ORR was 50.1% with the biosimilar and 44.8% with bevacizumab. The risk difference was 5.3%, and the lower margin was contained within, and the upper margin was outside the predefined equivalence margin. Additionally, in the full analysis set, the median progression-free survival was 8.5 months with SB8 compared with 7.9 months with reference bevacizumab, and the median overall survival was 14.9 months and 15.8 months, respectively. The median duration of response with SB8 and bevacizumab was 5.6 months and 5.85 months, respectively. In prostate cancer, treatment with relegilix achieved a high rate of sustained testosterone suppression to castrate levels in patients with androgen-sensitive advanced disease, meeting the primary endpoint of the phase 3 HERO trial. Findings showed that in the primary endpoint responder analysis, 96.7% of men receiving once-daily oral relegilix achieved sustained testosterone suppression to castrate levels from weeks 5 through 48. The data will serve as support for a new drug application to the FDA, which is anticipated to be submitted in the second quarter of 2020. Myovant Sciences, the developer of the agent, stated in a press release that the findings will also be included in future regulatory submissions in Europe and Japan. Prior phase 2 data also demonstrated efficacy with relegilix in patients with metastatic or locally advanced prostate cancer who were deemed appropriate for ADT. The results showed that the agent had a tolerable safety profile and that after 24 weeks, prostate-specific antigen levels were reduced by 97.3% to a median of 0.1 nanograms per milliliter, compared with 92.4% with a median of 0.2 nanograms per milliliter with loreprilin. The combination of nivolumab and ipilimumab did not show a statistically significant improvement in recurrence-free survival versus single-agent nivolumab as an adjuvant treatment for patients who have had complete surgical removal of stage 3 B, C, D, or stage 4 melanoma, and whose tumors express PD-L1 less than 1%, missing the co-primary endpoint of the phase 3 CheckMate 915 trial. The Data Monitoring Committee made a recommendation that the study continue unchanged. Additionally, the trial remains double-blinded and will continue to assess the other co-primary endpoint, which is RFS in the intent-to-treat population. Earlier findings with the combination demonstrated a 3-year sustained relapse-free survival benefit in patients with high-risk resected 3C4 melanoma. In a single-center 40-patient study, adjuvant nivolumab combined with ipilimumab led to a 71% relapse-free survival rate at 3 years. Single-agent nivolumab is also indicated for patients with melanoma and lymph node involvement or metastatic disease who have undergone complete resection in the adjuvant setting. Moreover, ipilimumab monotherapy is approved by the FDA for the adjuvant treatment of patients with stage 3 melanoma with pathologic involvement of regional lymph nodes larger than 1 millimeter who have undergone complete resection, including total lymphadenectomy, and also for unresectable or metastatic melanoma in adult and pediatric patients. The European Commission has approved pembrolizumab as a single agent or in combination with platinum and 5-FU chemotherapy for the frontline treatment of patients with metastatic or unresectable recurrent head and neck squamous cell carcinoma and PD-L1 expression. The approval is based on results from the Phase 3 KEYNOTE-48 trial, in which single-agent pembrolizumab demonstrated a 26% reduction in the risk of death compared with the standard extreme regimen, which comprises cetuximab with carboplatin or cisplatin plus 5-FU in patients with PD-L1 positive tumors. When used in combination with chemotherapy, the PD-1 inhibitor led to a 35% reduction in the risk of death. Both OS improvements were observed in patients whose tumors have PD-L1 expression. With the European approval, pembrolizumab as a single agent and in combination for this indication will be available for use in all 28 European Union member states, plus Iceland, Liechtenstein, and Norway. In multiple myeloma, the European Commission has approved the triplet regimen of daratumumab with lenalidomide and dexamethasone, known as DRD, for the treatment of adult patients with newly diagnosed disease who are ineligible for autologous stem cell transplant. The approval follows a positive opinion granted in October 2019 by the European Medicines Agency's Committee for Medicinal Products for Human Use and is based on findings from the Phase 3 MAIA trial. In the study, the daratumumab regimen led to a 44% reduction in the risk of disease progression or death versus lenalidomide dexamethasone alone in transplant-ineligible patients with newly diagnosed disease. At a median follow-up of 28 months, results also showed that the median progression-free survival for DRD has not yet been reached, compared with 31.9 months for patients who received RD alone. Moreover, DRD led to deeper responses versus RD alone, including higher rates of a complete response or better at 48% versus 25%. The overall response rate was also higher with the triplet regimen at 93% versus 81% respectively. The FDA approved the 3-drug combination of daratumumab, lenalidomide, and dexamethasone in June 2019 for use in this setting. This week, we sat down with Bruce E. Johnson, MD of Dana-Farber Cancer Institute to discuss next steps in research for BRAF mutant non-small cell lung cancer.
We think it's very important to define those patients who are likely to have a long response duration when they're treated with a BRAF inhibitor and a MEK inhibitor. We think there's likely to be some genomic or other characteristics within the tumor that we can identify that identifies those patients. The second thing we need to do in this BRAF population is to identify those who are more likely to respond to checkpoint inhibitors than to the BRAF and MEK inhibitors, because it's likely that there's going to be some of each in this group, because we know that some of the patients respond for very long periods of time while others progress in 6 months to a year. That's it, number one. Number two is that we need more potent and specific drugs. So we need better inhibitors of BRAF and better inhibitors of MEK. And lastly is we need to be able to attack different parts of the pathway. One of the things that we can think about is inhibiting the signaling through the RAF family of genes to develop drugs that inhibit not only BRAF, but the signaling through ARAF and CRAF, and also the ability to potentially inhibit not only MEK, but also the downstream MAP kinase pathways. The way I see things developing over the next several years is being able to go after different components of the pathway that BRAF mutants signal through and expand the number of kinases that we can attack, number one. Number two is being able, after those patients are no longer able to be effectively treated, that we know what agents to give them. So we want to know whether it's going to be helpful to give checkpoint inhibition plus continued tyrosine kinase inhibition, and to convert these people into people who are surviving for 2 to 4 years to surviving for 5 years or longer. The ongoing trial that I'd like to call people's attention to is being able to use encorafenib and binimetinib. The trial is just now opening and beginning to accrue, and we'll be able to compare the anti-tumor activity of those two agents compared to the already reported phase 2 trials of dabrafenib and trametinib. In addition, we are going to be seeing cohorts of patients treated with vemurafenib and cobimetinib, and we'll see how those particular patients do compared to the others. And lastly, as the information emerges about combining checkpoint inhibition with BRAF and MEK inhibition, we'll find out if that's applicable to the cohort of lung cancer.
That's all for today. Thank you for watching OncLive News Network. I'm Kristi Rosa.